Okay. Hello, everyone. My name is Muskaan Suvas, and I go to Rochester Adams High School, and I work under Dr. Gogoi. And this summer, I was given the project of looking at the effects of the loss of connective tissue growth factor, also known as Ctgf, in ovarian cancer cell lines. So ovarian cancer has a 5-year survival rate of about 50%, and this is due to its very quick, how quickly it metastasizes and how early on it metastasizes. And it's very hard to detect early on because of its vague symptoms. And as you can see on the pie chart, about 50%, 57% of ovarian cancer cases are diagnosed at the distant stage, which is when the cancer has already metastasized. And once metastasized, it can progress from early to advanced stages within a year, meaning it spreads throughout the body very quickly. Which is why our research question is, how can we better understand the mechanism of metastasis formation in ovarian cancer? For a little background, the process of EMT is when an epithelial cell transitions into a mesenchymal cell. And one of the main differences between an epithelial and mesenchymal cell is that mesenchymal cells are highly invasive, they're Noicus resistant, and they metastasize to other parts of the body. And during EMT, the epithelial cells undergo changes in the cytoskeleton and ECM, causing them to express traits of mesenchymal cells. All of these pathways contribute to EMT. However, we're going to be focusing on the Ctgf pathway. So Ctgf stands for Connective Tissue Growth Factor, and it is a key signaling and regulatory molecule involved in different biological processes such as cell proliferation, angiogenesis, wound healing, and more. And Ctgf actually has different effects based on the cancer it's in. In breast cancer, when Ctgf is overexpressed, it promotes cell proliferation and invasion. However, in ovarian cancer, when Ctgf is overexpressed, it inhibits cell proliferation and invasion. Which is why our hypothesis is that the loss of Ctgf acts as a trigger to start the process of EMT. And so in order to knock out Ctgf, the CRISPR-Cas method was used, in which a CRISPR molecule finds a precise location in the target DNA, a CRISPR enzyme cuts the target DNA, and a new custom sequence is added when the DNA is repaired. And in the R182 epithelial ovarian cancer cell line, the Ctgf knockout showed that the cells were more Noicus resistant and more invasive, which are both traits of mesenchymal cells. And this Western blot shows protein levels of Ctgf. As you can see, in the R182 Ctgf knockout, there is no Ctgf, and the same in the R2615, the Ctgf has completely been knocked out. And then in the R182 knockout, RNA sequencing was done, which is used to examine the quantity and sequences of RNA. And the I-pathway guide analysis was used to analyze the RNA-seq data in order to determine the most statistically significant. And these p-values show the most statistically significant pathways. You can see cell adhesion molecules and ECM receptor interactions are both pathways that relate to the ECM and cytoskeleton, which explains why we're looking at the changes in the ECM and cytoskeleton. And so that brings us to my first aim. My first aim is to determine if the Ctgf knockout reprograms the cytoskeleton of ovarian cancer cells. And I did this through immunofluorescence staining for lamellipodia. I did phalloidin staining for F-actin, which form microfilaments in the cytoskeleton. And the phalloidin directly binds to the F-actin and stains it green. And lamellipodia, as you can see in the diagram, are little protrusions along the edges of the cell, and their function is to act as a motor, which allows the cell to move forward during cell migration. And with lamellipodia, it's easier for cells to metastasize throughout the body. And RAC1 is an effective activated RAC1, which I'll talk about more later. This is the phalloidin staining for F-actin. As you can see in the R182, the cells are in their normal cuboidal shape. There are no protrusions along the edges. However, in the knockout, the cells are all deformed, and the arrow points to one specific formation of the lamellipodia. And the diagram next to it, the blue, shows DAPI staining, which highlights the nucleus of each cell. and the merged staining combines both the phalloidin, F-actin, and the DAPI staining into one picture. This is just a closer up version. As you can see, the arrow points to a formation of lamellipodia. And that is why my first Ames conclusion is that the loss of Ctgf reprograms the cytoskeleton, causing ovarian cancer cells to express traits such as lamellipodia, which lead to metastasis throughout the body. My second aim is to determine the different levels of expression of RAC1 between the wild type and the knockouts in order to understand its contribution to EMT and the reprogramming of the ECM and cytoskeleton. So what exactly is RAC1? Well, RAC1 is a transducer known to regulate multiple signaling pathways that control the cytoskeleton organization, transcription, and more. And when phosphorylated, RAC1 is actually deactivated and is activated when it binds to GTP. So this is the RAC1 pathway. As you can see, when RAC1 is bound to GTP, it activates the wave regulatory complex, which then activates ARP2/3, which then leads to the formation of lamellipodia. And so in order to measure the gene expression of RAC1, I did RT-qPCR, which has 3 steps. You start with denaturation, annealing, and extension. And I ran 2 qPCRs. As you can see, in the first one, the R182 knockout shows that the full change of RAC1 is a little more than 2. And the same is shown in the R2615 knockout. And our second qPCR showed similar results, meaning that in the knockouts, all the RAC1 change is upregulated, meaning that there's more RAC1 expression in the knockouts. And so once we validated the gene expression, we looked at the protein expression of RAC1 through Western blotting, in which a primary antibody binds to a specific protein, and then the second antibody binds to the primary, and then lets you detect the expression. So as you can see, our total protein expression of RAC1 in the Ctgf knockout is higher compared to the wild type. And however, phospho-RAC has lesser expression in the knockout. And this is because phospho-RAC is actually deactivated RAC. So if there's less deactivated RAC in the knockout, that must mean that there's more activated RAC present. And with that, our second Ames conclusion is that there's a higher expression of RAC1 in the knockouts in comparison to the wild type, and there's a higher level of activated RAC1, which would then cause the formation of lamellipodia. And so my overall conclusion is that the loss of Ctgf leads to a higher expression of total RAC1 and activated RAC1 in the knockouts, which would then cause the formation of lamellipodia due to the presence of activated RAC1 in the knockouts, which then makes it easier for a cell to travel and invade other parts of the body, leading to further metastasis. And some future studies would be confirmed that there is more activated RAC1 in the knockout of each cell line through an activation assay. What will be the effects of inhibiting RAC1 in both cell lines? Will inhibiting RAC1 in vivo slow down the process of metastasis? And how does knocking out of Ctgf lead to the activation of RAC1? I'd like to thank Dr. Moore, Dr. Gogoy, Dr. Alvaro, and the Discovery to Cure program for giving me this amazing opportunity. And I'd like to thank Sandra and all the lab members who helped me and guide me with my experiments. Thank you.